Hello, AP Bio, and welcome to Unit 7. Starting out with a little bit of humor here to introduce a couple of the topics in this unit. And while you're reading through all those hilarious comics right there, I just want to let you know your note packet for this unit is going to be a little bit off from how we go through the unit. Because of the changes in learning model coming up, I had to kind of rearrange some of the things that we're doing in this unit. So we're going through things in a little different order. Today's lesson is going to be a little bit of history about how we learned about DNA as the important molecule of inheritance and the structure of DNA. Most of this is the first few slides of your packet, though I added a little bit in, so you'll need to add some notes either in a notebook or on the sides of your packet. And then later in the unit, a couple big sections just kind of got flip-flopped. So I will try and remember to let you know kind of where we are as we move through the unit. But I just wanted you to know that up front, I did have to rearrange some things a little bit because of some of the upcoming changes. So this unit is on DNA structure and function. And so we'll start by talking about the structure of DNA. We'll talk about DNA replication. We'll talk about how the instructions of DNA are used to make protein and that process of protein synthesis. And then we will end this unit with some biotechnology, talking about some of the things that we're able to do with DNA and the uses that we have found for those technologies. So that's kind of where we're going over the next few weeks. We're starting with just the basic structure of DNA, which is section 3.6 in your textbook, and then moving into chapter 13, which talks a little more in depth about DNA and DNA replication. So if you want to reference the textbook at any point, that is where we are. So we're going to go through a little bit of history here. The purpose of this is not really for you to memorize every single name and every single date, but there are several really important experiments in the history of biology, and these experiments are often pulled up as examples in questions, or you'll see diagrams of these experiments. And so I want you to have at least kind of heard about these and understand their importance in the history of our understanding of DNA. I did put dates on these slides next to the scientists, just kind of for your reference, but I don't expect you to have them memorized. That's just so you can kind of place this in your mental historical story. So way back in about 1910, a scientist called T.H. Morgan did some experiments with fruit flies, and he was the first one who figured out that genes are part of chromosomes. People knew that there were these things called genes. You might remember learning about Gregor Mendel, who did experiments with pea plants and realized that something was carrying information from parents to offspring, and he called that thing, whatever it was, a gene. He didn't know the molecular structure of what he was talking about. He just knew there was something carrying information from parents to offspring. T.H. Morgan did some experiments with fruit flies and realized that those genes have to be part of chromosomes, because if you remember, during cell division, chromosomes are actually visible in cells. And by 1910, they could actually see chromosomes moving around, and they realized chromosomes were passed from parents to offspring. And so they figured those genes must be part of a chromosome. That narrowed it down a little bit, the search for kind of what this information holding molecule was. But chromosomes, the structure of a chromosome, and we'll talk more about this later, it contains DNA, but the DNA is wound around all kinds of proteins. That's the yellow in here. So they knew chromosomes were made of both DNA and proteins. So they knew that the genetic information had to be one of those. Well, what they knew at this time and over the next couple decades was that DNA was made of kind of four main building blocks, the C's, G's, A's, and T's. And it was usually pretty uniform in structure and had seemed to have similar types of functions. It was pretty much the same all the time. Proteins, on the other hand, had 20 building blocks. There are 20 different amino acids that can be mixed together. Proteins are very specific in terms of their shape. It's very specific to their function. And so just based on what they knew about these molecules, scientists at the time thought proteins were much more likely to be the genetic material because it seemed like it was a more complex molecule and a more specific molecule. And they thought those things would be important to something that held genetic information. But they weren't sure. It took several decades before scientists kind of answered this question for sure. And there were a few important experiments along the way. In 1928, Griffith did an experiment where he again just kind of showed that something was carrying information here. And what he did, you may have seen this experiment before, he had two different strains of bacteria. So what's labeled rough and smooth in this diagram are two different types of bacteria. The rough strain of bacteria could be injected into a mouse and the mouse would live. The mouse was fine. It was not pathogenic. It didn't make the mouse sick and kill it. But the S strain was very deadly. If you injected the smooth strain into a mouse, it would die. So he did a couple experiments with this. He took some of that smooth strain of bacteria and heated it up to kill the bacteria. And then he injected the dead bacteria into the mouse to see if it would still cause problems, but it didn't. The mouse lived. It was fine. So the heat-killed smooth strain was safe. The rough strain was safe. So then he took those two and he combined them together. So individually, the heat-killed smooth and the live rough strains both left the mouse alive. But when he combined them, the mouse died. And then when he analyzed that mouse, he found live smooth strain in here. And so his conclusion was that some bits of information were passed from that smooth strain into the rough bacteria and transformed them into that more virulent strain. He didn't know exactly what that was. He called it the transforming principle, but this was more evidence that there was something that could move between cells that carried information. Scientists still didn't know what that substance was. So in 1944, so a couple decades later, people were still trying to figure out this DNA or protein question. Avery and McLeod and McCarty did an experiment that kind of followed up on what Griffith had done 15, 16 years earlier. They took that same S strain of bacteria. So remember, this was the one that had killed the mouse originally. And they kind of figured out what was in here. And they said, okay, so bacteria contains DNA, it contains RNA, it contains lipids, and it contains proteins, and some other things. And so what they did is they took different strains and they mixed it with enzymes that would destroy that particular type of molecule. Remember, this ASE ending that you see on all these words refers to an enzyme. So for example, this one down here where it says protease, that's an enzyme that breaks down proteins. And they say, okay, if we break down the proteins in here and then they combine it with the R strain, is it still deadly? And it was. Okay, what if we destroy the lipids? Are the lipids the thing that's really making it deadly? If we get rid of the lipids, is it going to be safe? Nope, mouse still dead. What about the RNA? If we destroy the RNA, nope, still dead mouse. Okay, well, what if we destroy the DNA? And when they destroyed the DNA and broke up that DNA, then it no longer had the power to transform that R strain and the mouse lived. So they just kind of ruled out one by one all the different molecules that were found in that bacteria until they figured out the one that really needs to be there to make the transformation happen is the DNA. So this was the first kind of really big piece of evidence that DNA is the molecule that holds that genetic information. Hershey and Chase followed this up with another experiment in 1952 that also led to some really good evidence that DNA was the transforming principle. So here at the top, you can see this is a bacteriophage. A bacteriophage is a type of a virus. Remember, viruses are really simple in structure. They just have a genetic molecule. They have DNA or RNA surrounded by a protein coat. And that's really it. Viruses are very simple. And so what they did is they used phosphorus to label the DNA because phosphorus contains, or DNA contains lots of phosphorus in all the phosphate groups along the backbone, but protein doesn't. Protein contains a lot of sulfur. So in this one, they used sulfur to label the protein coat. They knew viruses injected something into a bacteria, and that something is what made that bacteria die. And they wanted to figure out what was being injected because they knew that would be the important molecule. So when they labeled the protein on the outside with sulfur, then they let the bacteriophage go and infect some bacteria. And then they kind of separated those out to see if that protein coat had been injected in the bacteria. They did not detect any sulfur in those cells. So they knew protein was not the material that had been injected. But when they labeled the DNA in the middle with phosphorus, then they went through, and then when they separated out the bacteria here, they did detect the phosphorus in there. So they knew that the DNA from that virus had been injected into that bacteria. So that was the information they were putting into the bacteria to replicate themselves, which in turn then eventually killed the bacteria. But that was further evidence that DNA, in fact, was the material that carried genetic information. So, of course, as this evidence started to pile up and people were starting to become more and more convinced that DNA was actually the molecule that mattered, people wanted to learn more about the structure of DNA. At this time, there hadn't been a ton of study done of the structure of DNA itself. So that was kind of the next focus of research. So if you get this joke, that means you already remember many of the important scientists who are involved in this story. As we tried to figure out, as scientists tried to figure out the structure of DNA, there were kind of a couple of important things. Sorry, I don't know why that says 1950 right in the middle there. But around 1950, the scientist named Erwin Chargaff had been studying DNA. He wasn't really too interested in the structure. He was more looking at the chemical makeup of it. And he figured out a couple things. By looking at DNA samples from a variety of different living things, he figured out that the composition of the bases, those A's, T's, C's, and G's, was different from one species to the next. So the composition you found in humans was different than in snails, was different than in trees. And he also found what is now known as Chargaff's rules, the second of Chargaff's rules, or the base pairing rules. He found that the percentage of A's, the adenine, was always about equal to the percentage of thymine, and the percentage of guanine was about equal to the percentage of cytosine. He didn't know exactly why that was important, but it was an observation that he made that held true no matter where he took his DNA sample from. In 1952, Rosalind Franklin, who was a scientist who was actively searching for the structure of DNA, took this picture that you see at the bottom of the screen that you may have seen before. She and Morris Wilkins, who was the other scientist who was working with her, were using X-ray crystallography, which is a way to kind of take pictures of how X-rays reflected off of a molecule. And when they saw this picture, they had this really clear X pattern in this picture. They knew from the calculations that they could do that that meant this molecule had the shape of a double helix. So they were really, really close to figuring out the structure of DNA. But meanwhile, at the same time, there were these guys, Watson and Crick. They weren't exactly doing a lot of like measuring or taking pictures or anything, but they had talked to Chargaff and they'd heard these rules and they had seen Rosalind Franklin's picture. They weren't really supposed to, but they saw this picture and they kind of put all these pieces together. And so Watson and Crick were the ones who successfully built the first accurate model of DNA. And this is actually what their model looks like. So those are kind of some of the key historical figures in the story of how we discovered the structure of DNA. What I would like you to do now, there are two things that I'm going to ask you to continue working on today after you finish watching this video. The first one, hang on, let me get out of here for a second. The first one, both of these are actually the first page in the Unit 7 Extras packet that you picked up from school. It's the one that had my little typed note on the front. The first page, if you turn it over, the back looks like this. there are a few practice problems that I'd like you to do using Chargaff's rules. So you're going to look at some actual data that was collected here and try and use Chargaff's rules to fill in the table and kind of explain how you can see Chargaff's rules in here. We're also using this as an opportunity to review a little bit of statistics. There are some things that you need to remember how to do from earlier in the school year. You may need to pull out your equations and formula sheet to help you with this, but work on this today. We will talk a little more about this page at the beginning of class tomorrow. But give it a shot now and see how far you can get. This is really important practice and things that you need to be able to do relating to Chargaff's rules. The other thing that I would like you to do tonight, kind of your homework for tomorrow, I'd like you to at least have this started by tomorrow. The front side of that worksheet looks like this. The chemical structure of DNA gives you a diagram. And then as we've done before, we're going to do a little bit of color coding and labeling. So at least try and start this part tonight. You do not need to worry about labeling the phosphodiester linkage. We'll do that part together because it's a little tricky to figure out exactly where that is. But you should be able to figure out these first 5. You can kind of color code them, describe any information that you find about them. Section 3.6 of your textbook will be helpful on this. And then there are a few more questions down here that you can begin to answer. This is the topic that we're going to talk about in class tomorrow. So please spend a little bit of time looking at this diagram and really studying what's in here so that you are familiar with some of the basic parts of DNA before you come to class tomorrow. Please email me. Let me know if you have any questions at all, and I hope you have a great rest of your day.